what I'm interested in now, or what I would like to present you, is post-translational modification of the GNE itself. Yeah? And the question behind is, why is there or how is GNE regulated and the enzyme activity could be regulated? And here you see an autoradiogram of GNE, which was incubated in vitro with protein kinase C, because GNE has several potential PKC phosphorylation sites. And we also did this, as you can see here, radioactive in the presence of an inhibitor of the protein kinase C and an activator of the protein kinase C. And what you see in principle, when you have no PKC, you have no phosphorylation of the GNE. In the presence of the PKC and the inhibitor, you also have no phosphorylation, but in the presence of PKC and no inhibitor, you have phosphorylation, and the stimulator PMA also increases it a bit. So this was the first data showing that GNE is phosphorylated or can be phosphorylated, at least in vitro. Some years later, we also checked another post-translational modification with this O-GlcNAcylation, and O-GlcNAcylation is very similar to protein phosphorylation because it occurs on the same residues like serine. And you see here the scheme, which is mainly introduced by Gerald Hart. So you have a naked protein and a kinase phosphorylated or phosphorylates a protein. Then you have a phosphoprotein, and phosphatases do the opposite thing. But the same protein can be also O-GlcNAculated by O-GlcNAc transferases, and also the O-GlcNAc can be removed. So this is a dynamic post-translational modification, such as the phosphorylation. And what you see here, at that time, we had no other possibility to analyze this, so we did an immunoprecipitation, and you see here the GNE, and you see here an antibody which recognizes the O-GlcNAc residue, and you see that the precipitated GNE is O-GlcNAculated. We repeated this several times, and in principle, we can now say, okay, GNE is phosphorylated and also O-GlcNAculated. Now I would like to introduce some data with the mutation, with the GNE myopathy mutation, and this is the M743T mutation, which is the founder mutation of the Middle East population. And this protein, or this GNE variant, was transfected in CHO cells and then analyzed in Western blot with different antibodies. And you see here the GNE antibody, and at that time we had not that good GNE antibodies as today. So you don't see the untransfected GNE in the cells, but the overexpressed wild type and also the overexpressed variant. And this was done by Dorit Bendmann in our lab. You see also that phospho antibody or phosphoserine antibody could detect the phosphorylated GNE. And what you also see here is that we get a O-GlcNAc signal, but the O-GlcNAc signal was much stronger in the mutated or in the variant, so in the Middle East variant, compared to the wild type. And this was just the loading control. So this was a hint to analyze this in more detail. And then Dorit did some, at that time we had the right radioactive substrate for the enzyme activity assays. Nowadays it's not anymore available, unfortunately, but we could analyze this. And you see now wild type and the enzyme activity of the epimerase. This is only the epimerase activity. You see here the activity of the wild type and the activity of the variant. And you also see that the variant activity is less but it's still around 50%. When these this cell supernatant or not supernatant, so these preparations were treated with phosphatase, then the, let's say, activity of the wild type was more or less unchanged, whereas the activity of the mutant or variant is a bit increased compared to the untreated or phosphatase untreated experiment. But what is very interesting is that using O-GlcNAcase, so the enzyme which removes O-GlcNAc from the protein, dramatically increase the activity both of the wild type. So the wild type is then higher than before, than untreated, and the mutated or the variant, so the Middle East variant, has nearly wild type activity. So post-translational modification, at least I suggest this here, has an influence on the activity, at least on the epimerase activity. The kinase was not analyzed at that time. Now I would like to come to another post-translational modification, and this is glycation. Glycation is a very interesting modification because it's non-enzymatic. So this is a chemical reaction, and most clinicians know this, maybe also some people who have diabetes here, because hemoglobin is a very important molecule which can be glycated, and the name for this is HB1C, so the glycated hemoglobin, and it's a sensor for the blood glucose level. And the reaction, the reaction is called Maillard reaction because of Louis Camille Maillard, who discovered this reaction. And it's a very easy reaction. So the amino group of proteins react with the carbonyl groups of sugars. This is in the first reaction a shift base, and then there are several further reactions, and then you have a stable compound or a stable post-translational modification, and the name for this is advanced glycation end product. And now I would like to go, this is glycolysis, so the way how glucose is metabolized. And I would like to draw your attention to the muscle, because the muscle is an organ where you have a lot of sugar metabolism, because the most glucose is stored in the muscle on glycogen, because this is a major, let's say, storage molecule in muscle. We have it also in liver, but at least most glucose is stored in the muscle. So you have a huge turnover on glucose in the muscle. And what I would like, so this is a normal glycolysis, and what I would like to show you here is that there's a side product on glycolysis, and this is methyl glyoxal. This is a normal, regular side product in the glycolysis. It occurs up to 1-2% in cells who do a lot of glycolysis. And methyl glyoxal has 2 carbonyl groups, and this is a very, very, very active molecule, and is 20,000-fold more active to form advanced glycation end products compared to glucose. So what we are interested then, of course, is GNE glycated or not glycated, because this is done by, the experiment was done by Elfun. She's sitting in, the picture was taken in Ottawa. But the question was now, do we have glycated GNE in normal cell culture? In normal cell culture means in high glucose. High glucose is normal cell culture medium have 4.5 gram glucose per liter, which is 25 millimolar, which is pretty high. And you see that we really have glycated GNE after immunoprecipitation. So this is an immunoprecipitation, and then we check glycation. And then you see when we have low glucose, it's a bit less glycated. And when we have high glucose and additionally MDO, which is this glycating agent from the glycolysis, we have higher, at least higher or greater amount of post-translational modification. And now coming to the last slides, Vanessa Hagenhaus could show in the lab, this is now published, that methyl glyoxal has a, let's say, weak effect on the activity on the epimerase. So you see here the epimerase activity after treatment with methyl glyoxal, and you see here the MANAC kinase, which is dramatically affected after glycation. So we have nearly no activity when the molecule is, or when GNE is glycated. We have checked this, also the glycation, so the molecule or the GNE is really glycated. So this is a, let's say, argument why you need certain time before you or you lose activity of the GNE, at least in cell culture. So this is a very short summary. There are post-translational modifications such as phosphorylation, O-GlcNAcylation, and glycation. And glycation is really important for the activity and also O-GlcNAcylation.